presentation. I will upload everything from last week and this week. Well, but maybe is this midterm season, that's why everyone is gone, right? Ah, yeah, that's a good point. I wish I can go somewhere. But somewhere where it's something not like a France, something where you can actually find the normal food, Tim Horton coffee.
Just
We-
do a staycation.
What's that?
You just stay wherever you want to stay, but go stay in a hotel and go to the pool and use the spa. And
That's what I will do on Saturday. Yeah, I'm going on Saturday, some rock. Hmm? What is
like a fancy older.
how much is that? How much? No, not going to happen. Haha.
The trick is, if you go on Hotwire, they have mystery hotels, and you can pick a mystery 5-star hotel for cheaper because you don't know which hotel you're gonna get.
Oh, so but you can end up in something like Econo Lounge when drug addict.
No, because if you pick your stars, they pick 4 or 5 stars. They'll have to be 4 or 5 stars because you don't know exactly what the hotel is. It's cheap.
How do you know that?
My dad travels a lot.
Oh
for
my
work.
God, I will need to take advice from you, really, because I'm horrible in picking hotel. Although I did try one very good hotel, it wasn't because I paid, so don't trick yourself. My salary is not big enough to cover that. Like that doesn't exist, but the hotel is on Lake Louise, like that. What's the Fairmont? Yeah, so again, don't trick yourself. Professor salary won't pay you that. Like that's not going to happen. But my best friend is traveling, like Marissa's dad, and because he travels for work, he gets those points on Expedia, and then he can use those points to book hotel, and then you pay maybe like so. I know how much is hotel. Hotel is like $1,700 per night, and I think he paid around 300 because he used his points for it. But two of us, and you actually have private bath hotel. Yeah, me and Richik, like my best friend, we're both like peasants. We really don't know what is that. So we didn't even know how to use all benefits. But I remember I forgot a swimming suit and I wanted to buy in hotel. I was like, I will just buy. There is $260 for like two-piece swimming suit. I'm like, not swimming. I'm just done. Like, even if I have $260, I wouldn't give it for this. Like, it's not even nice swimming suit. It's like nothing special, but it's in Fairmont. And there were people who bought it. But no, don't trick yourself. Not going to happen with this job. No. Like, I can lie to you and tell you, but it's not. So, anyway. But still, that doesn't matter because we love microbial physiology regardless of a low amount of money that you will get. Because money doesn't matter at all. You can always live in rented place. Or have a son who might play hockey and then you just got married rich and then I know that's what my mother told me 15 years ago, but I ended up I fall in love. That's a good concept. That's my mother told me literally the same 15 years ago, but it was like, honey, at least he is good. Okay, I'm kidding. No, really, I'm joking. Like, I'm really very romantic person, so I do believe in love, and I do believe in love for all life. So, and to love more, you can love more than once. So, it means if you got disappointed once, doesn't mean everyone is evil. No, it doesn't mean you can love more than once because there are nice people out there. So, microbial physiology. We are moving on with the harder part of early life metabolism because we covered the easier part last class. Now we are going to talk more in details about the Calvin cycle. Wait, it's not just Calvin cycle, the other. So we covered the reductive PCA cycle last like on Tuesday, and that's easy. Calvin cycle, not so easy. There are many enzymes that you have to know, and many important steps, and I will try to facilitate that now. So the most abundant and the highest in terms of energy efficiency, like the most expensive in terms of energy efficiency pathways for carbon dioxide fixation is Calvin cycle. And the whole function of Calvin cycle is to create trioses or 3-carbon sugars. And the reason why you have to have 3-carbon sugars is because these trioses are going to be used to build hexoses or 6-carbon sugar, which is a glucose. And glucose is a polymer of cellulose, polymer of starch, and those are all building blocks of plant cells. So, what Calvin cycle is doing, actually, enzymes in Calvin cycle is taking out the molecules of carbon dioxide straight from the air and turns them into the biomass, which literally means conversion of inorganic carbon, which carbon dioxide is, into the organic carbons, which are first trioses and then hexoses. So, what are steps of Calvin cycle? Again, the picture is there to give you a brief overview, but as usual, I write you everything so you can read it and then correct it. So what are steps? As we said, the molecule of carbon dioxide stays straight from the air. So the other molecule that you need the carbon dioxide to attach to combine with, is ribuloso 1,5-bisphosphate or RUBP, ribuloso 1,5-bisphosphate. So that is 5-carbon compound. And you would imagine once you have carbon dioxide plus this 5-carbon compound, then it's a 6-carbon compound. So that's the result. So initial first step of Calvin cycle. Next, from this 6-carbon compound, you are splitting it into 3-carbon compounds, which are 2 molecules of 3-phosphoglyceric acids or 3-PGA. And the enzyme that does that is carboxylase oxygenase or Rubisco. And you probably all heard about it. So 3-PGA molecules are converted into glyceraldehyde 3-phosphate. And this is the reaction that actually consumes energy. So consumes ATP and consumes NADPH. And those ATPs and NADPH are made in like reactions of oxygen. Again, we are now talking about fixation of carbon dioxide, which is done by Visco in that phase of photosynthesis. So the whole point is that from hexoses you get 3-phosphoglyceric acid. The 3-phosphoglyceric acid gets reduced to glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is one of the key molecules that you can use later on into metabolic processes. So one portion of glyceraldehyde 3-phosphate is going to be used to make glucose, which is carbon sugar. And another portion of glyceraldehyde 3-phosphate is used to regenerate RUBP or ribuloso 1,5-bisphosphate, which is 5-carbon compound. Why do you need to regenerate it? Because you have to have a molecule ready to accept a new molecule of carbon dioxide, so the cycle continues. So all process of Calvin cycle can be organized into mainly 3 steps. Like all the 6 to 7 steps that I mentioned can be divided broadly on carbon fixation, which are steps like 1 to 3, reduction, steps 4 to 5, and regeneration, which is step 6. So if you go back, this will be up to like when you get the phosphoglyceric acid reduction, and actually will be carbon fixation, then 4 to 5 will be reduction, and the 6th one will be regeneration. So carbon fixation, reduction, and regeneration are 3 phases of Calvin cycle. Why is Calvin cycle so energetically expensive? Because it consumes a lot of energy, which is then into ATPs. You don't have to really know how many, but you do need to know that it's really energetically expensive because of huge amount of energetic compounds like NADPH and ATP needed to reduce six carbon dioxide to get one capsule. Because you literally need six carbon dioxide. Now, two key enzymes from Calvin cycle would be Rubisco, and Rubisco is carboxylase oxygenase. Again, this is a S. And another key enzyme, we usually say this is second most important one, is phosphorylglucokinase. There are, and we will go in details what each of them is doing. There are other enzymes that are participating in Calvin cycle, like the reduction of 3-phosphoglyceric acid to glyceraldehyde 3-phosphate, and then glyceraldehyde 3-phosphate can go downstream to the metabolic pathway. But neither of these enzymes are very specific for Calvin cycle, so that's why I'm not mentioning any of this. Once when you have glyceraldehyde 3-phosphate, that one can go downstream may be converted to phosphoryl pyruvate, but then phosphoryl pyruvate was acetyl acetyl that the other stuff that is technically pentose phosphate pathway. And once when you have that molecule, you're good to go. Now, function of the Rubisco, and Rubisco again is very famous, and I mean, I'm pretty sure everyone heard about it, is to form two molecules of 3-phosphoglyceric acid from carbon dioxide and ribulose bisphosphate. It was one carbon. So the acceptor of carbon dioxide is ribulose bisphosphate. That is why this molecule needs to be regenerated so the process continues. The reduction of PGA, so the purpose of reduction of PGA is to make glyceraldehyde 3-phosphate. Because again, as I said, once when you have glyceraldehyde 3-phosphate, you can go wherever you want in the without. Phosphoribulose is second most important enzyme, and next to the Rubisco, so Rubisco is the first one. What phosphoribulose does, and again, the name tells you kinase, so that means it must add some phosphoryl group. So it takes ribulose 5-phosphate and makes ribulose 1,5-bisphosphate, like adds additional phosphate group, because that means it participates in creation or regeneration of acceptor for carbon dioxide. So Rubisco and the phosphoribulose are the most important. So what do you have in Calvin cycle is the need of 6 carbon dioxide molecules to make glucose, and you need 6 molecules of ribulose 1,5-bisphosphate that need to be regenerated through cycle. That literally means that you will spend 6 ATPs, which means 6 ATPs to regenerate ribulose bisphosphate, 12 NADHs and 12 ATPs to reduce PGA. And the problem, so very energetically expensive. The problem with Rubisco is that it is not only carboxylated, it also has oxygenase activity, which means it has activity and for carbon dioxide and for oxygen. And the result of Rubisco oxygenase activity is phosphoglycolate and 3-phosphoglycerate. 3-phosphoglycerate can be further converted. Phosphoglycolate is it's not really a full waste, but it takes an energy to be converted in photorespiration. So that's why we don't want that oxygenase activity. Again, photorespiration is the process needed to convert the result of Rubisco oxygenase activity, which is phosphoglycolate, and then 2 molecules of phosphoglycolate are converted to 1 carbon dioxide and 3-phosphoglycerate, which can enter Calvin cycle, but again, it spends additional energy. So you want to keep Rubisco carboxylate. In order to concentrate Rubisco and to protect it from oxygen, cell is organizing Rubisco and another enzyme called carbonic anhydrase in the compartment called carboxysomes. So they are literally, they're called pseudo organelles. So they are their own structures with their own transport system. So again, in plants, that's organelle. In cyanobacteria, they're called pseudo-organelles, but because they have their own transport system into the membrane, they technically are organelles, but they're compartments. That's why I call them compartments, because I don't like calling organelles in prokaryotes. And the role of carboxysome is to concentrate carbon dioxide and rubisco together. So it keeps enzyme and substrate in close proximity, and that's how it protects rubisco from oxygen. So it gets rid of oxygenase activity because there is no oxygen in them. So literally the place of carbon dioxide fixation, which is Calvin cycle, are carboxysomes which have hexameric structure. Another enzyme in carboxysomes is carbonic anhydrase, which is an enzyme that does rapid conversion between carbon dioxide in gas form to the carbonic ion, which is HCO3. Both bicarbonates and carbonates can pass through their pores, and the 3-phosphoglyceric acid can come out. So once the 3-phosphoglyceric acid gets out of carboxyl, it can be reduced to glycerol 3-phosphate. But the point is that these carboxyls have a membrane transport system that actually helps the concentration of carbon dioxide and its conversion to carbonic ion. Now, even though Calvin cycle is known to be the most abundant pathway, it is very energetically expensive, and it makes people wonder why this cycle evolved, since it's very energetically inefficient, why the oldest one and the one that is the most energetically efficient, Wood-Langdale pathway, did not evolve. And there is certain explanation, which you will tell me what do you think, do you agree? But the point is that Wood-Langdale is the ancient pathway for carbon dioxide fixation, which is used by acetogenic and methanogenic archaea, which are known as the early organisms on Earth, and this is very, very energetically efficient. So these acetogens or homo acetogens are organisms that have a very important role in ecosystems because they are consumers of intermediates from secondary fermentation. And what are intermediates from secondary fermentation? So gases like hydrogen and carbon dioxide. So just a brief reminder, primary fermentation products would be lactate, ethanol, or butyric acid. But then secondary fermentation products are gases like hydrogen, carbon dioxide, and homoacetogens, and later on you will see methanogens, are consuming and hydrogen and carbon dioxide, which are products of secondary fermentation. So they use hydrogen as electron donor, and they fix carbon dioxide using Wood-Langdale or so-called acetylcoenzyme pathway, and they excrete. So acetogens excrete acetate as a byproduct, while methanogens are excreting methane as a byproduct. So methanogenesis, or by methanogenesis, which results in production of methane, is done by organisms that we are known as methanogens. Acetogens is by acetogens. Both of them are oxidizing hydrogen as an energy source, and they fix carbon dioxide via acetylcoenzyme or Wood-Langdale pathway as their carbon source. Both of these types of organisms are extremely anaerobic. In fact, methanogens are known as the most strict, like the strictest anaerobes in this planet. And what it tells you is that these organisms were very early tenants of the Earth because Earth initially did not have oxygen in these guys. So this is the oldest version of metabolism. Some studies even say that it's older than anoxygenic water. So both acetogens and methanogens compete for hydrogen as an electron source. Another group of organisms that can compete with them are also like sulfidogens can also compete with them for carbon source, for energy source. And homoacetogens or acetogens are commonly studied in organisms like termites, ants that eat wood, and you will see why. So if you want to buy a prospect for these organisms, you buy a prospect in termite gut. These type of organisms have hydrogen abundant in the gut, and it consumes to So what termites are doing, they are consuming cellulose from wood. That's their carbon source. And in their gut, they are fermenting, like there are microbes that ferment that cellulose, and the product of fermentation is hydrogen. And because there is a lot of abundance of hydrogen as a secondary fermentation product, acetogens will live there because they consume hydrogen and they make acetate as their byproduct. So again, just what they're doing. So they're fermenting polysaccharides like the glucose, like actually cellulose. Secondary fermentation products are carbon dioxide. The hydrogen and carbon dioxide is all what you need all what you need for acetogen to make acetate because they use the Wood-Langdale pathway, and they said that enzyme hydrogenase that under. Now, so again, Wood-Langdale or acetyl coenzyme A, synonyms, same thing, and this pathway creates proton multiforce, like when there is electron transfer, there is a hydrogen. When there is electron jumping from carrier to carrier, there is a proton multiforce involved, and that multiforce is the electron used in biosynthesis, and carbon dioxide is fixed into the biomass. So all this, this acetyl coenzyme A pathway in acetogens has dual purpose. One purpose is to create your ion gradient to make it like transport of protons across the membrane. So while they're doing carbon dioxide fixation, so that proton multiforce is used to make ATP. And the another purpose of acetyl coenzyme A is to fix carbon dioxide into the biomass. So acetyl coenzyme A or Wood-Langdale pathway in acetogens has dual purpose: to fix carbon dioxide into the biomass, so that's a standard, and create ion gradient to make ATP. So step one, again, what we need to do first to get the carbon dioxide into biomass is fix it, like do some sort of carboxylation to create acid. Next one, you will combine acetate with coenzyme A, and you yield acetyl coenzyme A. That's why it's called acetyl coenzyme A pathway. And once when you have acetyl coenzyme A can go downstream and induce 4 or 5 reactions. So acetyl coenzyme A pathway is foundational pathway for all anaerobic life on this planet. And both acetogens and methanogens and animal organisms, like they are also using this one, are using this old pathway to fix carbon dioxide. Some organisms can create their own hydrogen through oxidation of pyruvate, because pyruvate is oxidated to acetate and the hydrogen and carbon dioxide byproduct, and then hydrogen and carbon dioxide are later on used to create acetate. And what is so this is just a broad overview of acetogenesis, and now we'll dig into the details how this goes step by step. So first of all, we call this acetyl coenzyme A cycle very often, but it's not correct. It's not a cycle. It's literally linear pathway, like 2 linear ways. But people commonly make mistakes, which is okay. What you have is one molecule of CO2 needs to be reduced to carbonyl group. Another molecule of CO2 needs to be reduced to methyl. Carbonyl and methyl group will give you acetate. So methyl and carbonyl group first. So you reduce carbon dioxide to methyl and carbonyl group. So they will assemble together and create 2 acetyl coenzyme A molecules. The key enzyme that will participate in creation of these 2 acetyl coenzyme A molecule is carbon monoxide dehydrogenase, or CO dehydrogenase. Then whenever you see the word dehydrogenase, that means there is some sort of oxygen reduction. So the enzyme carbon monoxide dehydrogenase is using electrons from hydrogen to reduce carbon dioxide. So you need dehydrogenase, carbon monoxide dehydrogenase, hydrogen and carbon dioxide. And then as a result, you get carbon monoxide and water. Carbon monoxide will be converted to carbonyl group of acetate. And now you have carbonyl, all good. And that was done by CO dehydrogenase. To make methyl, because you need methyl group as well, you will need assistance of tetrahydrocolor, which is a cofactor that works together with P12 containing with P12 containing enzyme. So it's enough to know coenzyme tetracolor. And then same enzyme, which is carbon monoxide dehydrogenase, will put together created carbonyl and methyl group and create acetyl. Like two acetyl coenzyme O and then acetyl. So again, not really hard. So hydrogen again. Hydrogen is the electron donor. CO is acceptor in this time, like it can be considered acceptor. So this is your pair. So this is oxidoreduction. What you have is CHO intermediate. Who is doing hydrogen oxidation? The enzyme that is very important for this process, present in acetogens, is the hydrogenase. It's we'll talk a lot about this enzyme in the next few slides. It's called the electron bifurcation enzyme. But for now, no, it's only enzyme that actually extracts electrons from hydrogen. So again, hydrogen is oxidized, it's taking out of hydrogen, then hydrogen. So what you need, of course, is participation of tetrahydrocolor to make methyl group. And again, carbon monoxide dehydrogenase is going to be the key. So why is this enzyme important? So you remember how I said that you have carbonyl and methyl group, okay? Then carbon monoxide dehydrogenase is participating technically in, well, not in methyl group formation, but it's important for final acetyl creation. So it is key enzyme that contains the, two most important metals, nickel and iron. So methyl group that is created by a cofactor tetrahydrocolor gets attached to the nickel enzyme of the nickel atom of enzyme carbon monoxide dehydrogenase. Then the enzyme is actually completing CO2 reduction and you get carbonyl group. But carbonyl group now gets attached to the iron. So you have an enzyme that attached methyl group onto the nickel and then performs reduction of CO2 and attaches carbonyl group. And now you have two groups hanging on the enzyme and they are in close proximity. And then carbon monoxide dehydrogenase forms the bond between carbonyl and methyl group. That's acetyl. Then you add coenzyme A and then you get acetyl coenzyme. And that's all what you need technically. The addition of coenzyme A is energizes, that addition of coenzyme A is energized by sodium multiforce. And that's something that we talked about when we talked about alkali files, like sodium multiforce. And that sodium multiforce is literally like sodium ions are flowing, creating a gradient through ATPase. And ATP is synthesized to generate more energy for the cell. But the point is that coenzyme A addition is energized by sodium multiforce. So, then you have that sodium multiforce that turns on ATP synthase. Now, you still need more ATP. And to get more ATP, you will have to do that now created acetyl coenzyme A hydrolysis. So you hydrolyze that acetyl coenzyme A to get more ATP because the reaction here is generating ATP, but as a byproduct, you have that acetate that gets excreted. So this is all for acetogenesis. What I wanted to mention briefly, and it's going to be important enzyme for the final exam, is this hydrogenase enzyme that is participating in hydrogen oxidation and energy conservation in acetogenesis. So energy conservation in acetogenesis occurs through this process called electron bifurcation, and the big role in that is played by cytoplasmic enzyme hydrogen. So that's the enzyme that actually extracts out this hydrogen. So as you know, what means oxygen reduction? Electrons are extracted. That means they are released, that means oxidation, something else that gets reduction. When electrons are extracted, protons are released in oxygen. But how you conserve energy is through that electron which is linked to this hydrogen. So what means bifurcation? So this is two-branch process, what hydrogen is doing. So when you extract electrons from hydrogen, those electrons have two routes. One portion of electrons will be used to reduce NADH and make NAD to make NADH, and the other portion of electrons is used to reduce pyridoxine compound, and then you get reduced pyridoxine. So that is the point of that electron bifurcation. That's possible with this hydrogen in there. So now you have two electron carriers, NADH and reduced pyridoxine. NADH reduced is used to create methyl group. You will have to have also that tetrahydrofolate cofactor. And reduced pyridoxine, again, ancient electron carrier, travels to sodium-driven pump to the membrane, and when it's getting oxidized, sodium ions are delivered to the membrane, and you get sodium multiforce created. This is called energy conservation through electron bifurcation. So literally, the definition of bifurcation enzyme, which hydrogen is the enzyme that is capable to extract electrons from one molecule, so there's one molecule, hydrogen, and use these electrons to reduce two separate molecules, which are NAD plus and pyridoxine. Why we need two of them to conserve energy? That means NADH will be used to the methyl group with participation of tetrahydrofolate. Reduced pyridoxine will participate into the turning on sodium multiforce because that's what we need. So, what's the role of acetyl coenzyme? Again, big and important role of energy conservation is linked to the acetyl coenzyme. So again, repetition. Hydrogen oxidized by bifurcation enzyme hydrogen. Carbon dioxide reduced, you have intermediate CHO. Tetrahydrofolate use this intermediate to make metal. CO dehydrogenase creates carbonyl. Methyl group remains attached to nickel of dehydrogenase. Carbonyl group remains attached to the iron of dehydrogenase. To just brief some. So dehydrogenase is the key enzyme of acetogenesis. It creates bond between carbonyl and methyl group. Remember, they now hang out on their metal atoms, and there is CO addition, coenzyme A addition, and you get acetylcoenzyme. Now, acetylcoenzyme can either be used for biomass, because it's a central carbon metabolite, or it can be hydrolyzed to make more ATP, and then you get a synthetic state. Now, methanogenesis is similar. It's a different type of organisms, but it has a certain connection with acetogenesis. First of all, methanogens are very strict anaerobic organisms. They are also as a homoacetogens consuming hydrogen and carbon dioxide, but they do not generate acetate, they generate methane. But some of them can consume acetate that acetogens made, so they can actually convert acetate to methane. Methane that is created by methanogens is consumed in oxic systems by organisms that we all are aware of, called methanotrophs. So methanogens live on the bottom in anaerobic areas, and the methane that they make goes from the top. And there are methanotrophs that live on the top, where there is oxic environment. So they, like, if we don't have those methanotrophs prevent methanogenesis release, how this planet will melt. So that's the only biogenic thing. Again, there are here no bacteria or eukaryotic organisms can form methane. So what do they contain? What methanogens contain? They have specialized enzyme for methylcoenzyme reductase. You'll see that later. And the enzyme is carrying carbon dioxide, so it's called C1 carrier. and it's needed to make methyl groups. And there are coenzymes M and B that are participating in redox reaction. So step by step, how that goes. So you again have hydrogen oxidized, and electrons are carried by hydrogen. So when hydrogen is oxidized, hydrogen, electrons are excreted, they are carried by hydrogen. Next step, you have methanofuran containing enzyme. So you will often see abbreviation MFR. And you need that MFR containing enzyme to activate carbon dioxide. Again, carbon dioxide is what you need to have to make methane. When you get reduction of carbon dioxide by this MFR enzyme, you will this time not get the carbonyl group, you're getting formyl. Formyl group is one of the differences. Formyl group can be attached to the methanoctarin containing enzyme, so you will see that as MPT. So again, this is a difference between acetogenesis because here you actually don't have carbonyl group, so you have formyl group attached to the MPT containing enzyme. And MPT containing enzyme is going to reduce that formyl group and form and methyl group is CH3. So now when you have that CH3, so you are one step away of methyl, which is CH4. So that's methyl group, CH3. So methyl group M, that's what we will remember, is going to be transferred to coenzyme M. That's what we will know. So coenzyme M gets accepted to methyl group. And then you will have, once a methyl group is on coenzyme M, you will have participation of this key enzyme MCR or methyl coenzyme M reductase that removes methyl group for coenzyme A, but MCR actually works together with coenzyme B, so it uses electrons from this coenzyme B cofactor, and it's reducing now methyl group to methane. So again, MCR and coenzyme B work together, and they literally take out methyl group from coenzyme M to make methyl. Not hard at all, right? That's why I had to make simplified version of this when I was studying it. So what are key players? Methyl coenzyme reductase works together with coenzyme B and coenzyme M, the one that actually has methyl. Coenzyme M gets that methyl group, and this is how it looks. So methyl group CH3 attached to the coenzyme M, and then MCR or methyl coenzyme reductase comes with coenzyme B. And then now that coenzyme B, again, alongside with this together with MCR, it forms, so coenzyme B comes with MCR and binds through SS bond with coenzyme M, the one that actually has methyl group. And it takes methyl group and MCR can reduce it. So that's all. So literally coenzyme B and MCR are working together in order to form methane. So if you roughly divide methanogenesis, again, there are three key players, MCR, coenzyme M, and coenzyme B. But if you roughly divide it, you can divide it in 4 stages. So first stage is the oxidoreduction, when hydrogen is oxidized, ferredoxin is reduced, and electrons are passed into CO2. And what you get as a form is formyl group, not carbonyl, as you had in acetogenesis. Step 2 will be passage of now formyl group to MPT containing enzyme or MPT, and there is a reduction of this formyl group to methyl group. And then stage 3 is when that methyl group now formed is passed to coenzyme M. And stage 4 is the final reduction, which is performed by MCR alongside with coenzyme B, when there is methyl formed and there is ATP produced. Again, methyl is byproduct. What about biosynthesis? So how we get molecules from biosynthesis? Again, you need acetyl coenzyme A. This is the key point. So the method that was made as a product is, so you have method as a product and you have generated the PNPM. But what you need to have for biosynthetic purposes, you need to have acetyl coenzyme A. So how can you get acetyl coenzyme A? Again, you need coenzyme A, you need methyl group. So methyl group comes from methane. You need also to have carbonyl group. Yeah, that's all for acetyl coenzyme A purposes. carbonyl group comes from same enzyme. So these guys also have carbon monoxide dehydrogenase. And this dehydrogenase is using that methyl group that comes from methanol and combines it to give acetyl at the end. Acetyl combines with coenzyme A, and you have acetyl coenzyme A for biosynthesis. So basically, the way how you get acetyl coenzyme A for biosynthesis is same in methanogen, also with participation of CO dehydrogenase and also methane, which means you need NCR and you also need to have coenzyme. So this pathway is the most energetically efficient. It's an ancient way. It does not require too many reduction equivalents of peridostin. The Calvin cycle, which is extremely expensive, like many ATPs manually. But Calvin cycle, the one that we covered initially, and the one that is the most abundant, is actually the most energetically expensive. So the question that people usually ask is, why would cell evolve such an energetically inexpensive pathway? Like why we would keep, why organisms would not just keep this the most energetically expensive wood long day? And the answer that people usually accept, and it makes sense to me, is that if you use it's literally about the molecules that you get at the end. So if you retain Wood-Langdale pathway, the molecules that you get at the end is acetyl coenzyme. Yes, it's great. Acetyl coenzyme can be further used for biosynthesis, but that will consume additional. Even though the pathway itself is energetically expensive, still you get acetyl coenzyme. With Calvin cycle, you get hexosis. So like molecules are more useful. So you get 6 carbon hexosis versus 2 carbon acetyl coenzyme. So I think that it's already accepted that hexosis is better molecule because it's a starting molecule for any purposes. But acetyl coenzyme, the 2 carbon molecule, must be built up to make any of the hexos, like any other molecule. So that's why Calvin cycle evolved and was retained compared to the one. Key enzymes, as we said, in rubisco and phosphoribosokinases in Calvin cycle, and these are enzymes linked to the PCA cycle. Rubisco can also be concentrated in carboxysomes. So again, those are all reasons to retain Calvin cycle versus the one pathway. So even though it's energetically inefficient, it is efficient from biosynthetic perspective. So that's why it is accepted as a main molecule. So we are literally done with the second lecture, and what we can do, can we see exams? Can I bring them?
Yeah.
No, I can. Okay, so because we finished earlier, I was wondering if you want to stay and see your exams. Okay, I didn't bring them because I didn't know how fast we finished. So I'm going, you wait for me here and I'm going upstairs to bring your exams. And then you can see it and we can talk about the questions that you have for